microplastic or how many microplastics. And English majors were having this, but I said, okay. So I just got rid of the S.
Yeah.
Thank you. It would have been a disaster if
No,
they had.
Maybe the
drinkers at the back can find a seat. Welcome. Thank you so much for coming out on such a nice summer's evening. Good evening. I'm Graham Shimield. I'm the director here at Bigelow Lab. Welcome to old friends and new faces for our Café Scientifique summer series. Great to see so many people here on such a nice evening. In a moment, I'm going to formally introduce Patty Matrai, but I'd like to give a little bit of advance notice for next week for the Café Scientifique, where we're going to talk about viruses. We're actually going to get to the smallest things in the ocean, and we're going to talk about how many viruses are in a teaspoon of seawater, and do they make a difference? And that's going to be presented by Joaquin Martinez, one of our senior scientists here. Do look on Facebook for the latest information about what's going on in the lab. And our next public tour, starting at 3 p.m. in the afternoon, is on Friday, August 12. So the end of next week. If anyone would like to see a little bit behind the scenes in the science labs and just get some idea of what's going on at the lab, Friday, August 12th at 3 p.m. So it's a great pleasure to introduce Patty Matrai. Patty's been with Bigelow a good number of years. I actually don't know how many. It doesn't really matter. 21. She started her scientific career as a Bachelor of Sciences at the University of Concepcion in Chile, and then went on to do both her master's and PhD at Scripps Institution of Oceanography in Southern California. Went on to a faculty position at the University of Florida, and she and Barney absolutely saw the light and came to Maine. I could tell you a lot about Patty's love of science on air-sea gas exchange and the Arctic, and how environmental change can be measured by using some of the latest technology. But instead, I want to stress a couple of other things about Patty's approach to science, which includes collaboration, particularly internationally, and working with some of the groups that are trying to look to the future of what this planet is going to be like, what it means for mankind and society. And she works tirelessly in those areas, as well as in the professional organizations that represent scientists in this country and abroad. So quite beyond what she brings in terms of her own science, she works tirelessly on behalf of the scientific community and sort of society in general. And for that, we're extremely grateful. So she's taken that challenge on tonight in quite a big way with this title. I'm sure it's going to be thought-provoking for you as the audience, and we're looking forward to hearing your questions as they come up through her presentation. And without further ado, we're going to find out all about plastics, microplastics. Thank you, Patty.
So, welcome to Bigelow. First of all, I have instructions about the square footage here where I can move in. So I'm just taking, okay? I have 2 pieces of tape. Those, as Graham said, for those of you who have heard me talk before, microplastics is not something that I have done before. It actually came to be as a challenge from a member of the audience. I'll just put it as that. And then a challenge from some at the lab while I said, yes, of course I will talk about microplastics. And when we eat seafood, and my heart sunk, okay? Because I am definitely not a seafood expert. I am a lover of seafood. I eat every possible type of seafood, some things that you probably wouldn't consider. So today's presentation is not only the result of my investigations, but they actually represent almost all of the data in here is the work of my summer intern, Stephanie Peart, from Stockton University in Jersey, who has spent the last 10 weeks with me. And I'm going to ask her to stand up. Because if you have any difficult questions, you please direct them to Stephanie, okay? There also was extensive bibliographic research. And here I have to acknowledge our librarian, another member of the public, and this young lady, Maya McGuire from the University of Florida, for whom 20 years ago I was in her PhD committee when she did her PhD in corals at the University of Miami. And she was instrumental in helping me put together what I'm going to do today. So, with that, the very first thing I did was change the title, of course, okay? And so I am going to focus on micro nanoplastics in the marine food web. Because in order to get to seafood, it's not really a lot of the seafood that we ingest that is ingesting. Some of them are ingesting microplastic directly, but most of it is indirectly through the food that they ingest, okay? So some definitions to begin with. First of all, can you hear me all the way into the back? Okay, I don't know who does this. Can you up the volume a little bit? Because otherwise I will just, okay? That's not better? Okay. So microplastics are defined as pieces of plastic that are less than 5 millimeters. Well, we don't really do metric in the US. And so 5 millimeters is the width of the words liberty in a penny. Right there, okay? So now everybody bring out their penny and you can see exactly what I'm talking about. Small. But it is not microscopic as we would use in science. It is not micron. It actually should be milliplastic if we want to do measurements, okay? So that's the first thing. And there are two types of microplastics. Primary, those which are made of that size on purpose. And secondary, those that result from the fragmentation of many bigger things. And here I'm going to stop because this is the only time you'll hear me in this entire talk about large plastics. And I have to honor the work of many people and many agencies, organizations all over the world who are counting, cleaning plastics, beaches, sediments, oceans, everywhere. I'm not going to talk about that. There will be no pictures of seabirds or mammals or any charismatic megafauna whose health is being imperiled by the presence of plastics, okay? We can perhaps skim all those out. It is what happens to those plastics once they get degraded, destroyed, cut up, however it is. And Stephanie and I can tell you that it's really difficult to get a plastic cup and make lots of little pieces out of it. Difficult, very difficult. That's another story you can ask afterwards. Okay? And so within the microplastics, within the primary ones, folks talk about nurdles and microbeads. Nurdles are the original stalk, if you wish, from which all of these things are then become smaller. It may be that they are in your pillow, accent pillow somewhere, or in those ones that some of us who travel wear around our necks when we are on an airplane, okay? Microbeads tend to be in cosmetics of all kinds of sorts, from toothpaste to facial things. Secondary microplastics are the fragments of all kinds of things. But there is one type that I actually was very surprised as to how numerous they are, and those are fibers, okay? That come mostly from our clothing and from carpet. They are everywhere, right? Anybody here has worn a fleece, and then you go and it's just everywhere. We don't even think about it. Most of it is in sheds from fabrics during laundering. Very difficult to count, very difficult to isolate as well. And so why do we care about these microplastics? They are in the environment, and they seem to be increasing in abundance. And I'm going to stop here again. Plastics, there is a societal value to plastics. All of us here today are wearing plastic of some sort and type. In our clothes, our shoes, our watches, our hearing aids, our pacemakers, our knee replacement, our earrings, you name it. The chairs that you're sitting on. There are lots of things that plastics are very good for. But it's the excess, is the trash, is the stuff that we throw out. It does not belong in the ocean. It does not belong in the environment. That's what's increasing in abundance. Does not biodegrade very easily. Yes, I know, I have biodegradable plastics in my kitchen as well, but they degrade at temperatures that don't exist in the oceans. We're getting better perhaps. There are certain toxins that may adsorb to their surfaces. As you will see a little bit later, it's difficult to know how much. That they stick to it, yes. They're eaten by marine life, and that there are all kinds of additives that they have that can actually leach into the tissue of organisms. But I live and breathe at Bigelow. And Bigelow's mission is to investigate the microbial drivers of global ocean processes. And so we'll start at the beginning of the food chain, which starts with phytoplankton. And so with Stephanie's help, this is now Stephanie's research, we looked at the impact of microplastics on phytoplankton growth. Is there any? That's concentration matter. That's a type of plastic matter. Are different species impacted in the same way? And so we selected 3 different types of very common microplastics. Polystyrene, such as what's in styrofoam cups. Polyethylene, probably the most common one in the oceans. Cosmetics. Acrylic ester, something that we find in paint. We selected 3 different species, a diatom, a coccoleurophore, and a chromist. And we selected 3 different concentrations. And this is also very important. 1,000 per milliliter is what's measured out of wastewater treatment plants, okay? 1,000,000 beads per milliliter is an equivalent 1-to-1 to the natural concentrations of phytoplankton cells during a summer bloom or harmful algal blooms, okay? But the concentrations which are being measured and reported in the seas are 10 to 100 per liter, okay? So that's at least 1,000 times less than what Stephanie and I chose to use. So you could say, well, Patty, just go home, right? Because everything that you're going to say is not going to apply to the environment. But if there is bioaccumulation, if there are perhaps hotspots, we always start with the bigger effect, and then we start to go down to the lowest and lowest effect. So keep that in mind as I go through the entire talk, okay? So that's concentration matter. And you actually are going to get data today, okay? And so what you have here, we culture a diatom. This is its growth rate, how fast it grows. The higher the number, the faster, the better, the happier it is. There are 3 different treatments that I just described, low, medium, and high, okay? And this is a picture of this diatom, 3 different shapes, and we used polystyrene for this particular experiment. We, royal we. I waved, she worked, okay? And so what you see here is the reds are the experiments that had microplastics in the cells. Everything else is a control because we didn't know what we were going to find, right? So the very first thing is you grow the cells by themselves. That's the green bar. Then we discussed, well, might these microplastic beads make it darker? or they're white, maybe they reflect light, maybe they make it lighter. So we also did a light control, which is the blue bar. And then when you, if you've ever had these little pieces of plastic in your clothes, what happens? They stick, right? And so they're highly hydrophobic. And so they usually come in a soapy solution. Well, so what if it's just a soap, right? So you have a soap control. And so the only thing that you need to remember is that, look, these are all pretty much the same, except for this one. So at the highest concentration, this diatom grew more. What did we expect? Of course, I wanted it to crash, right? That's what I expected it, to grow less. But it's not light, and it's not the soap. So park that one there. What is it? I don't know at this point. That's the type of plastic matter. So we grew the same diatom, but now with 3 different plastics. The orange bars are the same data that I just showed you. We also used polyethylene, which are the ones which are in cosmetics. And we used acrylic ester, which is what's found in paint. Again, you have controls over here. And if we focused on the red ones, which is the polyethylene, you see that, especially this one, it's very high at this medium concentration. But at the highest concentration, which is this bar here now, it was happier. It's extremely unhappy. It crashed completely. So we have the same diatom, the same concentration, 2 plastic, and we have opposite responses. So I said to Stephanie, you did something wrong. Sorry, dear. Okay? You got to repeat the experiment. And so she did, with a different species. This is Coccoliophore. And she saw the exact same response. Okay? So they don't like that plastic at that very high concentration. Or something that is on the plastic, I don't know. And so the third one is, do different phytoplankton species respond the same way? And I've already given you a hint that perhaps they do. Here's the data that I showed you at the very beginning with the 2 controls and then being happier at the highest concentration. Sorry, with the 3 concentrations, happiest at the highest concentration. This is in the Carteret, where at the highest concentration, this is a different experiment in this particular case, in a different plastic. This is the same plastic as the very beginning, the polystyrene. There's really no difference. So for this plastic, it doesn't seem to care. This is Rhodomonas salinus. Oh, I have pictures of them. That's right. So here's the diatom. This is a little small one, very preferred as food by many invertebrates. And here's the Coccoliophore. It doesn't like it as well. Its growth is reduced at some medium concentration. So I have 3 different species. Do they respond the same way to this plastic? No. Earlier I showed you that 2 responded the same way to a different plastic. So, does concentration matter? Yes. Does the type of plastic matter? Yes. Are different species similarly impacted? No. And we did this experiment in the lab under controlled circumstances, conditions, 1 at a time. But that's not what happens out there. You have a community of phytoplankton, all different, all at different growth stages, all with different physiologies and different ecologies. How does Stephanie scale up from the lab results to what's happening out in the ocean? Lots of question marks, okay? And so I'm going to stop here because I will change pace a little bit afterwards and take questions from you at this point from what I have presented. No questions in a room with 70 plus people? Oh my God. Yes.
When you were showing the impact of different types of plastics, I didn't follow the acronyms. Which types of plastics inhibit the growth the most and which the least?
So from what we have here, this one here, which is polyethylene, which is, if I go all the way back to, is what we find in cosmetics, okay? That, for now, seems the one that has the most negative effect. But life is not always negative. Polystyrene actually encouraged growth. So if some of them, their growth is encouraged, and we put a lot, continue to put more of this type of plastic, might then those grow more, faster? What does that do to the composition of the phytoplankton community? So if you don't start asking any questions, I'll start asking questions to you. Yes?
So the size and the shape of the plastic that you use is exactly the same for each type of plastic?
Yes. They were all spheres, and they were all within a range of 40 to 64 microns. Yes, Kirsten?
Is there any data on which of the compounds that you tested is the most abundant one, like the
So the question is, are there any data as to which of these plastics is most abundant in the oceans? Thousands of data points. They're not beautiful. They're beautiful from the perspective of the data available. They're very sad from the perspective of the abundance. The most abundant is polyethylene, this one, which is the one that is related to microbeads. Yes, ma'am.
What medium did you grow them in?
We grow them in? In L1. Okay, all right. So with that, so medium is effectively a soup. It's filtered seawater to which you have added back extra nutrients in the right proportions that mimics what's in seawater, such that they will not be limited by any of the nutrients. Over time, over a period of 10 days, by day number 10, they are running out of some of those nutrients in the particular type of experiment that we did. Yes. Yes, sir.
Is your assumption that this increased growth rate is a good thing?
I'm not going to say that. I don't know enough. Because good is a relative term. It may be good for that species, but it might be bad for the grazer. And so from a perspective of community, microbial community, that's a very difficult statement to answer. Is a harmful algal bloom good? Well, it is for those phytoplankton, but not for anything else.
We're talking about bioaccumulation only.
Yes, but the phytoplankton I have not seen, nor anybody. Sorry, I repeat the question. Sorry. The question is, are we ultimately talking about bioaccumulation? And my answer is, there is no one who has yet measured bioaccumulation in phytoplankton. Okay? So I'll take one more question somewhere in the back, and then I got to go. Yes, sir.
What is the impact of variable ocean temperature?
I'm sorry, can you repeat that?
What is the impact of variable ocean temperature?
So the question is, what is the impact of variable ocean temperature? Forget plastics, but just like all living creatures, we grow faster at slightly warmer temperatures, and we grow slower, we, including us, humans, at colder temperatures. And so in this particular case, all the conditions, temperature, light, were constant for everything. So I eliminated that out of this current experiment. So hold your questions, okay? So then now we have the phytoplankton, and they're going to be preyed on by zooplankton, the next level up in the food chain. And I have, if my, oh, there it is. And so I found this beautiful, oh, I don't want the music. Go away. Okay, so here you have zooplankton feeding on all of these fluorescents or fluorescent microbeads of about the same size as microalgae, phytoplankton. And what you see are, as they feed normally, they're not discriminating. And so the fluorescence allows you to see it. This is, I think, it looks maybe like a ctenophore. Never mind all the ads. Okay, I'm not doing product placement here. And so you see them inside, they're tracked, okay? And so if I now go back to my own talk. And here I am showing data from another intern here at Bigelow who's not present, Brian Kim. He was an intern in January as part of the collaboration between Colby College and Bigelow Laboratory, an extension of the semester program that we run. And what he did in collaboration with David Fields, who was his mentor, another one of the senior scientists, he raised 2 different types of copepods on Rhodomona salina. That's one of the species of phytoplankton that we tested as well, and fluorescent microbeads. And so when he compared the ingestion, per copepod per day, which is the rates that you see on the y-axis, in the 2 copepods, the red bar with the diagonals is the phytoplankton. The green one with the little dots are the microbeads. And so what you see, well, there's no difference, Patty, right? That implies that they were just as happy taking in the microbeads as they were taking the algae. Okay? And so then what happens to that? What's the repercussion of that? Did it stay inside their gut? Well, the only way to know if it stays inside their gut is to measure what comes out. And so there is a very big interest in fecal pellets in the oceanographic community, because fecal pellets, oh, interesting, something has happened, which I call sinker or floaters here. It is the most important, the fastest way of getting carbon or biomass from the surface of the ocean or the upper layers to depths, to the sediments, to the benthic organisms, to the mussels, to the oysters, to anything that we might eat as part of the seafood that filters, okay? And so what you see in these pictures here is fecal pellets that have a mixture of algae and microbeads, fecal pellets that are mostly only of microbeads, and then the brown ones just with algae. And the red symbols are the fecal pellets full of algae. The green ones are those filled with beads. The ones with algae sink faster. The other ones sink slower. Okay? In other words, they're not getting down to those. Fecal pellets are a source of food for all kinds of things. And so they're not getting there. And they excluded out of this type of fluorescent green. Those actually floated instead of sinking. Okay? So there is the potential for a disruption of this, what we call benthic pelagic coupling. So do filter feeders excrete plastics after they filter them out? Yes. What impacts do they have on marine microorganisms? Oof, that is what I'm trying to give you a feel for. can these nanoplastics bioaccumulate in the microbial food chain? I will show you data which is not mine that suggests so. And there are potential human health risks. We're getting there. And so here are several reports in the literature and highly reputable journals within the last 2 years. Here is an oyster, and Monahan reports that there are these microplastics which are hurting decreasing oyster reproduction. Now they were able to measure rates of reproduction. And you will hear me use the word rates a lot now. It's not enough to know how much or how many are there. You need to also know how they're affecting change. Do they grow less? Do they reproduce less? Do they prey less? Okay? Here is a report in Science again of a fish larva, and I don't know if you can see, but there's got all kinds of little beads that it has eaten and appears to influence the ecology of fish larvae. Now we all eat fish. No, I'm not sorry. Many people eat fish. There's a report, again this year in Scientific American, where they looked at microplastics in table salt. And it's there, okay? Now, I'm telling you that in this case, they're present. Does it do anything? We don't know. Are we consuming it? Yes, we are. Here is one where they found plastic debris, microplastic in fibers in fish and bivalves sold for human consumption in the US market, in Indonesia as well. And so what do we do? No, is there any way to degrade these plastics? And I don't know why this is doing this, but it is. It says biological degradation on plastic. And this is a report by engineers in Stanford of mealworms. Now, this is on land, in which they're capable, if you see all these little holes, of apparently eating plastic, okay? Is there something like it in the oceans? Don't know yet. Earlier this March, there was a report in Science or Nature about bacteria on land being able to degrade plastics. And so, might this happen into the ocean? In 2013, colleagues from the Sea Semester program in Woods Hole, who is one of the largest groups, they've collected and sampled microplastics all over the oceans. They have the largest, probably, data set. And what they found, you see all of these round, these are bacteria that are living in the pits of the plastics. So the obvious question is, did they go into the pits or did they create the pits? We don't know that. All they report is that there are bacteria living in the plastics. If they're living in the plastics, which are naturally buoyant, that's why we see them everywhere, well, perhaps this is going to make the plastic a little bit heavier. Might then they sink? And this is one of the things that we find now. There are actually microplastics over the entire water column, all the way into the benthos. But when we look at abundance in the water column, not at the surface, which is what 99.9% of the microplastic samples are collected from, or in the sediments, we don't have almost no data, okay? So I can stop here for a few more questions, because I'm now going to change topics a little bit as to how has society done to deal with this, okay? So I'd be happy to take a few more questions. Yes, sir?
Microorganisms that are consuming plastic, are they altering it through the digestive process, or are they excreting the plastic?
So the question is as to whether organisms which are consuming plastics in their organism, is it degraded or changed in any way? We have almost no data. Because for that, you would need to know what the composition of the plastic was before it got into the gut of the organisms. And there's going to be, I'm going to use the word, I almost use the word natural, but environmental photodegradation, for example, of some of those plastics. That happens perhaps before it even gets to the gut of the animal. So we don't know. I hope that there is a chemical engineer out there who is devising this unique method that will allow us to answer that question. Okay? Yes?
Could you tell us if there is an adaptive or evolutionary response that you're seeing among the organisms that are probably stressed by ingestion of microplastics?
So the question is whether we know that there is an evolutionary response by the organisms that are ingesting plastics. We barely have any data about organisms ingesting it. We don't have any data about the rates at which they're ingesting it. Okay? So, no, we don't know. Okay? Yes, and then I'll move on. Yes?
How much of the testing that you're talking about done at laboratory level and the laboratory conditions? How much in the open?
All of it is done at the laboratory level. Okay? And so, I don't know why this is happening. Just stay there. How much plastic enters the ocean? There is an enormous amount of work being done on quantifying how much plastic there is. There is a very recent study by Jambeck et al. In Science 2015, and they took the data from 192 countries. And this is a very clear way of understanding it. It's the equivalent in 1 year, 5 grocery bags per foot of shoreline. Okay? And so if we look at the number of microbeads estimated to enter the waters in the US daily, okay, 8 trillion. So these are very big numbers. And earlier, the gentleman right here in the front row asked me, are you going to scare me so that I cannot eat any fish anymore? And I said, no. Because these numbers are enormous. They should not be in the ocean. No questions. But when you look at the volume of the ocean, you end up with actually something like 19 to maybe 100 particles per liter. Per liter. That means that there is less than 1 particle per milliliter. And Stephanie and I used, in our low concentration, 1,000 particles per milliliter. And at those concentrations, we didn't really see much of an effect. Okay? Now, are these low numbers? This is if I take these numbers and divide them by the entire ocean. You have to understand, people did not go measuring the entire ocean. They only took a few places, and then you scale up. Okay? But by the same token, you can scale down. So this is the big question. What can we do to recover this plastic? Well, it's a big challenge. It's small. Now, it's not just at the surface of the water, as I said earlier, because perhaps you could take some large skimmer and then just filter all of the water from the surface. But now we see it over the entire water column. And how do you remove the plastics without removing the marine life, the phytoplankton, the bacteria, the zooplankton, the viruses that you will come to hear next week, right? Removing them at the same time that you remove the plastic. These squares are 3 millimeters on the side. And so these are tiny. And so what can we do? First of all, we have to recognize that there are societal benefits for plastic. But there are no societal benefits for the plastic refuse, the trash that we generate. And so we can reduce, reuse, recycle, refuse. You can check the labels for the products that you buy. You can participate in beach cleanups. You can look for the very small plastic, but beware, don't be lulled that because you've gone and picked up 25 plastic bottles or cups, you have actually done something for these microplastics. they're going to be too small for you to see. And so there are a variety of companies who have agreed to get rid of the microbeads over the last 2 years. There is local and state legislation from several states, including Maine, San Francisco, Washington, D.C., who are changing their laws and banning what's called single-use water bottles. And the most significant one was the passage in December of 2015, so just last year, of the Microbead Free Waters Act of 2015, which will be effective in 2017, outlawing the use of microbeads. And so I brought you to the world of micro and nanoplastics and microbial food webs. Let's go back to the original title and let's look at what we do have and what we don't have. So we have a lot of data on plastics abundance. We have less data on microplastic abundance. That's at the millimeter scale. We have few or no data on what I call nanoplastic abundance. That's at micron. That's the size of all the microorganisms in the ocean. We need systematic methods. Every person, every institute, every country is measuring them in a different way. So it's very difficult to compare the many different studies. With the new methods, how do you tell which plastic it is? Somebody asked me. And so then we need data on all of these rates, growth, ingestion, bioaccumulation, morbidity or mortality, degradation, toxicity, right? That's the big question. That's what you all came here to hear me say. It is toxic. I can't answer that question, okay? So many unanswered questions. There was recently a discussion covered by the EPA, 2014, published in 2015, in which they concluded that the current state of the science does not allow an assessment of possible human health risk from the ingestion of seafood contaminated with all kinds of things. And furthermore, that there is no way to compare the results that are coming from microplastics to other sources. We get the same toxins that comes in dust, that comes in from runoff water, that comes in from a variety of other sources. In order for one of us standing up front to say, this is bad, you also need to compare to everything else that is coming in, okay? And so, if I bring it back to Bigelow, and this is my last slide, we have the Center for Venture Research on Seafood Security. And what the center, the CVR on seafood security is doing is bringing together the people, the institutions that work on seafood security in order to engender science-based solutions, even if it's not us at Bigelow who are creating them, that will result in informed policy. And the third goal is already being met by having you all here in order to raise public awareness. And with that, I'll be happy to take any questions. Yes, sir, in the back.
Has there been any attempt to actually measure plastic concentrations in seafood itself?
Yeah. So the question is, has there been any attempt to measure plastic concentration in seafood itself? Yes. In mussels, in crabs, in some fish, in worms, even though we don't quite, most of us consider worms seafood. So there is. It's present.
Okay. Is it present at levels that are of concern, or is it?
How much is it concern? So you need to do the study on the human side in order to determine what is concerned as well. If it's only the plastic, if it is the toxins which are with the plastic, then you would need to determine what's the threshold that is of concern for humans. Some of those studies have been done for PCBs and DDTs and all of that. But what we don't know is, do the plastics really carry that much? That's actually a lot more difficult than we think it is, especially if we only have 10 to 100 particles per liter. That's cubic meters of water that you need to filter in order to get enough of a sample. Yes, sir.
Years ago, they were talking about the problem of mercury in fish. Is that the same problem, or can it be handled in the same way?
So the question is about mercury in fish. And mercury in fish is something that the state of the toxicologist of Maine pays an enormous amount of attention to that. It is mostly for freshwater fish, though there is some also on marine fish. Marine fish, there is a natural source of mercury. So in marine fish, it tends to be less of a concern. In freshwater fish, it continues to be a major concern. Microplastics could be and should be studied in exactly the same way. Same protocols, same. For that, you need to have everyone using the same methods so that you can compare the studies. Yes, yes, sir. And then I'll take over here.
Follow up on the question this gentleman asked. Have there been any experiments done where you raise fish in an environment that has one level of microplastics versus raising fish in an environment that has another level of microplastics? Feed the fish to a test animal and see if there are adverse health effects?
Not to that extent that you're taking it. The study that I showed here with the fish larvae is one of the first ones to look at fish larvae exposed to microplastics. The transfer over the food web is something that is just beginning. And by that I mean both in the US and in Europe, where they're actually far more advanced on this type of studies than here in the US. There's a dent here.
Do you have any sense of how long it will take for enough data to accumulate for the EPA to make a more definitive statement about
You give me the money, I'll do the research. So it is an issue, I'm being very serious. It really is an issue of focus, of funding, and of priorities. You are all here. Clearly for you, for me, this is very important. It just gave me the shivers, the question of the gentleman, because the last thing I ever imagined was doing work on microplastics, okay? All I see are the big charismatic megafauna with actually very low effort to look at transfers across the food chain and all of those things, because those are not as sexy as cleaning the beach or saving the seal. Forgive me, okay? Microorganisms are not as sexy, but they're significantly more important. Without them, whether it's the human microbiome or it is the marine microbiome, we wouldn't be here. Yeah? And so that's really, so what can you do? Put pressure. Yes, sir.
Another question, you mentioned that the concentration of microplastics in ocean water was 10 to 100 per liter. And you then mentioned other quote-unquote bad stuff that gets into the water, dust, PCBs, and things like that. What sort of concentration do you find of other bad things in a liter of seawater? In other words, is 10 to 100 an infinitesimal concentration or a high concentration?
I'm going to hire you. Come work in my lab. That's the attitude that I want, okay? That's exactly what you have to think of the big picture. Do we know what the concentrations are in sea?